Welcome to the lecture on hormonal regulation of metabolism. Here are the lecture objectives telling you how to take from this the details you need. Really great place to start for your study. We start with the concept of metabolic homeostasis. And homeostasis just simply means what the status quo is, what's going on in the cell at any given time with the metabolism in the cell. And so cells are going to need a constant supply of fuels in order to make the ATP that's necessary for whatever process they're responsible for. Muscle contraction, nerve transmission, DNA synthesis, protein synthesis, whatever. So let's consider glucose. Glucose is the universal fuel, and we'll talk about the glycolytic pathway coming up in a couple lectures. For an average person, an average adult, about 190 to 200 grams of glucose is needed every day by that person. And of that glucose that's needed, most of it, 120, 150 or so, is used for the brain alone. So you see the brain as a major consumer of the glucose that we need to have on a daily basis. The remainder is going to be used by other tissues. Now you don't have to memorize this, don't memorize this. But what you recognize is that we have to have glucose, and the brain is going to consume most of that glucose. So the way we accomplish this is that our blood is going to hold that glucose. There's going to be a certain amount of glucose in our blood at any given time, and that is about 80 to 100 milligrams per deciliter. That's the normal amount of blood glucose that your average person will have. And if you have that level, there's enough glucose in the blood at any given time to maintain those metabolic processes and keep the cells running the way they're supposed to. Now, if the glucose drops lower than, say, 60 milligrams per deciliter, there's no longer enough glucose in the blood to drive those cellular processes. And so somehow we have to signal to the system that we need more glucose in the blood. We need to get that concentration up so the cells can take that glucose in and start doing the cellular processes. That balance must be achieved. And so there's always going to be a balance between carbohydrates, fats, and amino acids that are used as fuel sources. Carbohydrate or glucose needs to be used by those tissues that absolutely must have glucose. And then fat can be used by tissues that could use fat, but they could also use glucose. And then same thing for amino acids or protein. And so the different tissues will say, well, I can use glucose when the level is high, but when the level is low, I can switch to fat and spare the glucose for some other tissue. So the question is, who makes that decision? Who tells the body when there's enough glucose in the blood or not enough glucose in the blood? Or who tells the body when there's too much glucose in the blood? And who tells the body when it's time to start using fats or amino acids? And that's going to come down to that metabolic homeostasis and these hormonal signals that help us control that homeostasis. So the strategies for maintaining blood glucose is going to be local control, where we can change the concentration of metabolites or we can change enzyme activities. For example, if you need more fat to make more energy, then adipose can release more fat, and that fat will be more substrate, and those enzymes have greater substrate, greater activity, and those kinds of things. Okay, that's fine. That's all that enzyme kinetic stuff we talked about before. But the other strategy we can use would be more global, and that would be using certain hormones to carry certain messages that each tissue can understand and tell that tissue what's going on and how to respond. And it turns out that global strategy is a better way for us to maintain homeostasis. Of course, locally, the heart or the brain or the liver will have different enzymes that are more or less active and different substrate levels and that kind of stuff. But hormonal control is really going to be the key to carry that message throughout the entire body. And the two signals that we have, the two messengers, are insulin and glucagon that we mentioned before. Insulin is going to talk about blood glucose, and when the blood glucose level is higher than normal, that's when insulin is out doing its thing. And then when we're fasting, when the blood glucose is at the normal level, that is where glucagon is going to be in charge. Their job is to maintain that blood glucose at or near 100 to 80 to 100 milligrams per deciliter. Both of these hormones are going to be synthesized in the pancreas and released from different cell clusters in the pancreas here. Insulin is the pancreatic beta cells, glucagon is the pancreatic alpha cells. Now, these are the only hormones that are released in response to circulating levels of fuels in the blood. And when we get into physiology, when you get into digestion and you get into all these different hormonal signals that affect digestion and release, okay, that's fine. Those are the organs, but we're talking about in the blood metabolic hormones here. They're also called counter-regulatory hormones because each hormone is going to act in a way that is opposite to the other. So insulin signals the high glucose, blood glucose state. Glucagon signals the need for more glucose into the blood. Okay. And we call insulin the boss hormone because glucagon is pretty stable most of the time. It's always there, it's always doing its thing. But then as blood glucose levels rise, insulin levels rise, and they turn you into an insulin metabolism. When the blood glucose comes down, the insulin level comes down, and glucagon is still there doing its thing. So I like to call insulin the boss hormone. But they are always working, they're always in balance. Okay, let's take a look at insulin here. It's a major anabolic hormone of the body, which tells all tissues that glucose is available in the blood. Typically, 80 to 100 milligrams per deciliter is the basal level of glucose, and tissues recognize that. And some tissues say, thank you, I've got to have that glucose. Brain and red blood cells will, and so that basal level of glucose is going to be good enough to keep those organs happy. What insulin tells these tissues is, insulin says there is more glucose available than the basal level, and so you can take that glucose up and you can use it. And so insulin will tell liver to take up glucose and use it for its own energy needs. It tells adipose to take up glucose. It tells skeletal muscle, all muscle, to take up glucose and to use it. Okay, that's the signal that insulin is saying. Again, brain and red blood cells, they don't care about insulin. They say, I take up glucose anyway. You don't need to tell me there's extra glucose. I'm going to take glucose up anyway. But these other tissues that have reversible metabolism, fat or glucose, are going to hear that insulin signal. Now, it's produced by the pancreatic beta cells, and when insulin binds to the cell receptors, the insulin receptors, it's going to turn on all those signaling pathways that allow the cell to take up the glucose, use that glucose, but also store the extra glucose or to make other molecules that can be stored as well. If you're a liver, you're going to make glycogen. If you're an adipose tissue, you're going to be storing fat. If you're a liver, you can make fat and export that to the adipose, and then you can store that as triglyceride as well. And again, we have lectures coming up for that. Now, the release of insulin is going to be dictated entirely by the blood glucose level. And that's really important to go down here and see this. We saw this picture before. This is the hexokinase enzyme and the glucokinase enzyme. They have the same function. They take a molecule of ATP, they take a molecule of glucose, and they will phosphorylate that glucose to make glucose 6-phosphate, and then the other product is ADP. Hexokinase is the enzyme that exists in most tissues like skeletal muscle, brain, red blood cells, and hexokinase has a very, very low Km, which means it has extremely high affinity for glucose. So even at very low levels of glucose in the blood, that glucose gets into the cell and gets phosphorylated so it can be used by the glycolytic pathways. Glucokinase is the enzyme we find in the liver, and it's what we find in the pancreas. Glucokinase has a much more allosteric look to it, that S-shaped curve here, that sigmoidal curve. And what you find is this curve comes up much slower. It takes much higher levels of glucose in the blood to start stimulating this enzyme to become more active, and you very slowly rising your activity as you try to approach the Vmax over here. So glucokinase has much lower affinity for glucose, and the enzyme is much less responsive to the glucose. So why is that important? Well, if you're a brain or a red blood cell and you depend on glucose, you want to have a very high affinity hexokinase enzyme to get that glucose phosphorylated and get it into energy production. But the liver says, I'm happy using glucose, I'm also happy using fat. And so I'm going to wait and see just how much glucose is out there. So as the blood glucose rises higher and higher after a meal, as more glucose is absorbed into the blood and the glucose in the blood gets higher, you see the liver enzyme will become more and more and more active. And if you have a little bit more glucose, you have a little bit more activity. If you have a little bit less glucose, you have a little bit less activity. But still, hexokinase, the littlest bit of glucose, is going to stimulate that enzyme to Vmax very rapidly. Now, what about the pancreas? Well, the pancreas is not shown in this figure, but it's the exact same thing. The pancreas says, my job is to monitor blood glucose, and if I find blood glucose levels going up higher than 100 milligrams per deciliter, I need to start releasing insulin. Well, how does the pancreas sense that? Well, there's going to be glucose in the blood, and the glucokinase enzyme now is going to be governing this phosphorylation in the pancreas as well. So when the blood glucose level is low, there's really not a lot of glucokinase activity, but as the glucose level gets higher and higher and higher, the glucose that gets into the pancreas will also become phosphorylated more and more and more based on how much glucose is out in the blood. So why is that important? This is the process by which pancreatic cells sense glucose. So here's a pancreas, pancreatic beta cell right here. Glucose will come into the cell and it will turn on glycolysis, and glycolysis leads to pyruvate and TCA cycle and oxidative phosphorylation, and that allows the cell to make the ATP it needs to do its work. And so the ATP level will rise as this glucose comes in. But the sensing mechanism exists out here on the plasma membrane. As the ATP levels get higher and higher and higher, there are specific channels that will bind that ATP. Right here you see a red negative. That means the ATP is going to bind to this potassium channel and cause it to close. And when that potassium channel closes, potassium doesn't go out anymore, and that's going to change the voltage of the cell. And when the voltage of the cell changes, you see a green plus here. And when that when that voltage change affects this channel, calcium is allowed to come in. And when calcium comes into the cell, the concentration inside the cell gets higher. And that calcium now will bind to specific machinery inside the cell that will cause insulin, which is stored inside this cell, to move to the plasma membrane, fuse with the plasma membrane, and release it. If you remember, calcium is high outside of cells, calcium is low inside of cells, so as soon as that channel opens, calcium will flood in and very rapidly increase the concentration, which allows that to happen. So as long as the blood glucose remains high enough to support ATP production that's high enough to keep this potassium channel closed, then insulin will be released. But as soon as the insulin goes out into the blood and cells start to take up that insulin, the blood glucose level will fall. And eventually the glucose level will fall low enough that the ATP production will slow. There's no longer a high enough ATP level to cause this potassium channel to stay closed, so it will open back up. And when the potassium channel opens, the voltage goes back to normal. When the voltage goes back to normal, the calcium channel over here will close. When that calcium channel closes, there's no longer calcium to stimulate insulin release, and the insulin release will fall off over time. So it's a beautiful, beautiful mechanism that works. And later on, we'll talk a little bit about diabetes, and this mechanism right here is very much perturbed for in diabetes processes. There's about 15 different things that go on in diabetes, and this is certainly one of them. Okay, so what is the regulator of insulin release? The major regulator is going to be glucose. According to this picture here, glucose goes in, ATP levels rise, and you kick off this sequence here. So glucose is the major signal that controls insulin release. But you see there's some other minor regulators as well. Amino acids can give you a little bit of insulin release. Your brain can tell the pancreatic beta cells to start releasing a little bit of insulin. And then there are gut hormones, they're called incretins, that we'll see a little bit later, that can also change. Now, epinephrine is going to be a major negative modulator, and we'll talk about the fight or flight response. We talked about it in signaling, but we'll talk about it in terms of metabolism soon. During the fight or flight signal, you want glucose to flood your blood, you want to flood your tissues, and you want that glucose to stimulate ATP production as rapidly as possible. And so the last thing you want is for insulin to be released and cause the insulin to make the glucose go to all these different cells. And so epinephrine will tell insulin to cool it because the glucose is needed for the fight or the flight syndrome here. We'll talk a little bit later about how amino acids can stimulate insulin release. We'll talk about that coming up. It's not major. It's not like blood glucose. If you have a piece of bread, white bread for example, you're going to have a massive insulin release. If you eat a bit of protein, you'll have a small insulin release. And so there is insulin release, but just the magnitude is what matters. And just to remind you, we're going to go through all of these pathways, but just to remind you that insulin binds to the insulin receptor, and there are a number of adapter proteins that are bound, and those adapter proteins will become phosphorylated, and kinases become phosphorylated, and calcium signals and stuff. There's a lot of stuff that goes on, but the main thing that happens is protein phosphatase 1 is going to be activated to undo any phosphorylated proteins that happen by the glucagon signaling. We saw that back in the cell signaling lecture. Here's protein phosphatase 1 that when it gets activated, it's going to control the phosphorylation state of any proteins that are there, phosphorylated proteins. Okay, how about glucagon? Glucagon's that catabolic hormone. It tells you're in the fasted state. Glucagon tells adipose tissue to release fats. It tells liver to take up fats for energy. It tells liver to produce glucose for the blood. Okay, so these things are going on. It's telling this body that there is no fuel molecules coming in from the diet. You have to rely on stored fuel, stored glucose, stored fat in order to meet the needs of the body. So glucagon levels typically will increase as insulin levels start to fall. Yeah, it's a little bit more, we'll see a nice picture coming up here, but insulin is the one that goes up and insulin is the one that comes down, and glucagon is fairly stable over time. Let's see that. Sorry, one more slide after that. So glucagon is synthesized and secreted by pancreatic alpha cells, and here's a molecular picture of what it looks like there. The major regulators here, of course, glucose. Glucose is going to stimulate insulin, and insulin, being the boss hormone, is going to cause a negative effect on glucagon release. Insulin and glucose together will have a negative effect. Now, amino acids have a positive influence, and that is coming from the fact that during fasting, with the pools of amino acids we have and amino acids going into the blood, those amino acids can help stimulate a little extra glucagon release to help the liver take that amino acid up and use it to make glucose. Minor regulators are going to be cortisol, the stress hormone, when you have actual stress mechanisms in the nerves, and of course the fight or flight. These are things that will send glucagon out because glucagon is the hormone that signals release energy from the store, release stored fuels and get them out into the body so they can be used to make energy. And as a reminder about how glucagon works, it's a G protein coupled receptor. Glucagon binds, G protein's turned on, adenylate cyclase is turned on. That's where you get the cyclic AMP. Cyclic AMP will activate protein kinase A, and protein kinase A will have a number of proteins that are targets in the cell. And when the protein gets phosphorylated, that will change its state. And so typically glucagon and insulin will work counter-regulatory to each other. What glucagon turns on, insulin will turn off, and what glucagon turns off, insulin will turn on. So it comes down to the fuel availability and whatever the tissues need, whatever their demands are. And this is really a simple picture, but it's the right idea. Based on what is needed by the tissue, the body will say, do I have any fuels coming in from the diet? Have I eaten recently? Am I still absorbing them? And if not, I've got to take them from the fuel stores. I've got to take them from fat and liver and those kinds of things. And so after a meal, when a person is fed and the insulin level is high, the insulin is going to say there is glucose available. You can take this glucose in and use it for your own needs or whatever. Okay. During fasting, though, glucagon is in charge, and there is not insulin in the blood, and so that tells the liver you can't take glucose in, but instead you need to be producing glucose, and you need to send that glucose out into the blood for the tissues that are going to need it here. And the difference between these two, of course, is going to be the pancreas. When the pancreas senses higher than normal blood glucose, it releases insulin. When the pancreas recognizes the blood glucose level is normal, it's going to release glucagon. And that is the way these two hormones are going to be sent out and how they're going to work together. If you were to look at this picture over here and say, is this going to be fasting or is this going to be the fed state? Well, what's going on? Here you see the liver is producing glucose. It's actually sending glucose out, and the brain is going to be the major customer. Down here you see adipose is sending fatty acids out. so those fatty acids can be used by the liver and by the skeletal muscle. And so right away you would recognize this must be a fasting figure, because the liver is sending things out and adipose is sending things out. And in fact, glucagon tells you that right away. Glucagon is what causes these changes to take place. All right, this is one of those really, really important figures that will help so much with so many courses along the way. And this is showing you how does our body respond to a high carbohydrate versus a high protein meal. So if we start over here on the left, we're going to be looking at a person consuming a high carbohydrate meal. And what that bar is right there is that period of time that the person is consuming the food. It's getting in their stomach, getting in their digestive tract, and it's being absorbed. So prior to eating the meal, which is where this line is, prior to eating the meal, you see the blood glucose level is going to be at 80 to 100 milligrams per deciliter. The person is in a fasting state. They have not yet eaten their meal. But as soon as they eat the meal, you notice the blood glucose level starts to rise because those carbohydrates are being absorbed into the bloodstream. And as the blood glucose level rises, the blood glucose is being sensed by the pancreas, and the pancreas will release insulin in response to the rising blood glucose levels. And as the insulin level comes up here and gets to a peak, you see the blood glucose level will start to fall again. During this period of time, the meal has been consumed, it's in the digestive tract, it's being absorbed. Where the bar ends tells you that's when all that absorption is done and all that blood glucose is in the blood now. And so very quickly the blood glucose will fall because you're not bringing any more glucose in from the digestive tract, but rather you're absorbing the glucose out of the blood and into the tissues. And as the blood glucose falls, the pancreas says, I recognize the blood glucose level is getting smaller and smaller and smaller, and so the pancreas will release less and less and less insulin over that period of time. So it's a very much interdependent process. Well, what's going on with glucagon over here? Glucagon during fasting was in control, pretty high level here. And then during a meal, you see a little bit of a spike here because our body doesn't know exactly what's being eaten. It doesn't know if it's protein or fat, or we'll talk about that a little bit here. But as the blood glucose level rises, as the insulin level rises, you remember that glucose and insulin are both major suppressors of glucagon. And so as insulin and glucose rise, glucagon will fall. And glucagon is going to sort of hang out down here for a while until the insulin level comes back down. And eventually you can see that little uptick here. It's starting to come back up where it needs to be over here. Now what's important is look at the scale here. Here's 80 to 100. That's the basal level of blood glucose. After a high carbohydrate meal, this level goes up much above the 100 level right there. So your blood glucose is significantly higher than the basal level, which is why you're getting insulin released. Notice here that you're going from near zero, a very low level of insulin during fasting, to a very high level at the height of absorption of that blood glucose. And then you're going to work your way back down to a near zero. Over here, notice your scale. You're looking at a very small change, whereas this is 0 to 120. Look at this. This is like 90, 100, 110, 120. So while this looks like a huge drop, it really isn't that big of a drop after all. It's just a very small suppression of the glucagon. We'll see another picture soon that shows glucagon is fairly flat over time. All right, what happens if you have a high protein meal? Same scenario, the person is fasting, everything is flat because you're fasting, and then the person will consume a high protein meal and really no carbohydrates. So during this period of time, the protein is being digested, the amino acids are being absorbed, and it's getting into the blood. And you see there's really no significant change in glucose here because the person is not consuming carbohydrate. They're not consuming glucose, so the blood glucose level really doesn't change. During fasting, the liver's job is to maintain that blood glucose level at 80 to 100 milligrams per deciliter, and it's obviously doing its job here. So when a person eats, we'll see this coming up here, we'll see it a little bit here, there is neural factors involved. The brain recognizes that we're eating, and the brain is going to send a signal to the pancreas to have a little bit of a release of insulin so that it's almost like a precursor to the digestion and absorption. So you'll always see a little bit of a spike here, but look at the scale, 10 to 20 micro units per mil here. Over here we were looking at 120. So that's just a little tiny little blip. But what you'll notice is because the blood glucose doesn't change much, that insulin that gets released falls off really quickly because the pancreas says, I don't see any change from the basal level. I don't see any need to have a large release of insulin. I don't see any need to have a sustained release of insulin. So that goes away pretty quickly. And when insulin is down, glucagon is up, and now you're seeing this robust rise in glucagon because glucagon says, you know what? We have amino acids coming in, we don't have glucose coming in, we don't have insulin present, so that means I'm in charge and I'm going to be driving the process. And one of the things glucagon will do is it will stimulate the amino acid pools in the blood and stimulate using amino acids as carbon skeletons to help give the liver what it needs to make glucose. And because of that, we see that nitrogen in the blood is going to rise during the hours that follow the high protein meal because you're using more amino acids for metabolism and you're going to be producing more free ammonia that has to be disposed of. And that's why that nitrogen line is going up. So when we compare the two, these are really, really great cartoons. They're so simple and they're so effective. Under an insulin signal, insulin says there's more than enough glucose in the blood. Everybody can have glucose. Everybody can use the glucose to meet their own energy needs. Everybody can use the glucose to do whatever they need to do in the cell. So if you're a liver, you're going to take the glucose in and you have plenty of glucose. So in addition to meeting your own energy needs, the first thing you want to do is you want to synthesize glycogen to get the stored form of glucose to full. And if you have excess glucose over that, you're going to use the glucose to make fat. And then you can export that fat for storage in the adipose. And we'll talk about this process coming up. Okay. If you're an adipose tissue, you're going to say, wow, there's excess glucose. I can meet my own energy needs, and I bet there's a lot of fat out there that's going to be stored. And so you use that glucose not only to meet the energy need, but to make the materials that are necessary to build triglycerides and to store them inside the adipose tissue. And of course, if you're a muscle then, the muscle says, under an insulin signal, I recognize there's more than enough glucose out there. I am happy to take a lot of glucose in. I can build up my glycogen stores, so I have a little bit of stored glucose during times of exertion. And I can also use that to meet my immediate energy needs as well. And of course, amino acids are going to be stimulated to come in because skeletal muscle needs to constantly turn over its protein and synthesize proteins and so forth for the liver as well. Liver will suck up amino acids because liver makes a lot of proteins that are sent out into the blood for a variety of blood processes. Then when you're in fasting stage, it's just the opposite. Now you're under glucagon. Under glucagon, the liver's job is to put glucose out into the blood. So what you find is the glycogen, which is the stored glucose, is going to break down to make free glucose for export. Because there's not excess glucose anymore, you're no longer going to be synthesizing fatty acids, you're no longer going to be synthesizing glycogen, but rather you're going to be putting that glucose out. When the glycogen stores run out and the liver has to start synthesizing its own glucose, it's going to use amino acids as a precursor, so those amino acids will be stimulated to come in as precursors for the glucose. Notice that glucagon tells adipose tissues to break down triglyceride, send the free fats out. It's not shown here, but the glycerol will also be taken up as a precursor for glucose synthesis. And then over here, you see the muscle says no effect. Muscle is very happy to use fat. It uses fat all the time. It doesn't need glucagon to tell it to start using fat. It will use fat all the time. The difference is when glucose is available and insulin is present, insulin will tell the muscle that there is an excess of glucose, and it will stimulate the muscle to take up more glucose under the insulin signal. So let's look a little bit about this reciprocal regulation idea we talked about before, and let's talk a little bit more about insulin and glucagon and how it turns on and turns off specific pathways. And glycogen is a great example. We've seen this already. We saw this in the cell signaling lecture, and so we'll talk about it here in more metabolic terms. And if you recall, glucagon is going to be that hormone that binds to the receptor, the G protein, adenylate cyclase, cyclic AMP, protein kinase A. So protein kinase A, when it gets activated by cyclic AMP, it's going to have two major effects on glycogen metabolism. The first thing it's going to do is it's going to stop this synthesis. Glucagon is signaling the fasted state. Glucagon says there's not enough glucose out in the blood, there's not an excess. And liver, you need to start producing glucose and sending it out to the blood. So the liver says, I don't want to use my glucose to build glycogen, but rather I want to break glycogen down to get glucose out in the blood. So you stop synthesizing it, and then you turn on glycogen degradation down here. You want to start breaking down the glycogen, you want to start getting individual glucose units, and you want to start sending them out to the blood. So the net result of cyclic AMP activation of protein kinase A is going to be favoring glycogen breakdown, liberation of glucose, and glucose going out into the blood. So you see that here, for example. This is the signaling pathway that we saw before. This is during glucagon signaling. Glucagon binds to the receptor. There's the G protein. The G protein talks to adenylate cyclase. Adenylate cyclase makes cyclic AMP. Cyclic AMP turns on protein kinase A, which is now active. And that protein kinase A is going to phosphorylate proteins and turn them on. And one of the things that happens is, here's the glycogen synthesis process. So the glycogen synthase was active, but when protein kinase A got turned on, it phosphorylates the glycogen synthase. And when the glycogen synthase is phosphorylated, that's a negative allosteric change on that protein. It becomes inactive. And so glycogen synthesis will essentially stop. Over here, when protein kinase is active, it's going to turn on another kinase, the phosphorylase kinase. We've talked about that, and we'll talk about it in the glycogen lecture again. But this leads to the phosphorylation of glycogen phosphorylase enzyme. That's the enzyme right there that will break down glycogen. And glycogen now is going to go to individual glucose units, which can then be processed and released into the blood. So the same signal, glucagon binding to the receptor, turns off glycogen synthesis. It turns on glycogen degradation here. Another thing that happens is, because there's a low level of glucose in the blood, the glucose transporters are not very active. There's really not much glucose coming into the blood, and that's going to diminish any phosphorylation of glucose and any feeding in of the, utilizing glucose as an energy source. Of course, insulin undoes what glucagon does. So when you have a large high carbohydrate meal and you have blood glucose, we go to this next one here, it's going to have an opposite effect. It's going to lead to the activation of the glycogen synthase. It's going to turn off the glycogen degradation process. And the main way it does that is through the dephosphorylation by protein phosphatase mechanism. When protein kinase A is active, protein kinase A is phosphorylating those protein targets, and that phosphorylation changes their activity. But then insulin activates the phosphatase, and the phosphatase takes that phosphate off of those proteins, turns their activity back it was before, and the state turns back to that insulin state here. Okay. So let's see. So I use the old picture here with these X's to show you what happens. After a carbohydrate meal, and glucagon is no longer in charge, the glucagon receptor is now quiet, the G protein is quiet, the adenylate cyclase is quiet, cyclic AMP levels are going to be low because the phosphodiesterase is going to degrade those. When cyclic AMP levels are low, the protein kinase A is no longer active. And when protein kinase A is no longer active, it can't keep phosphorylating the synthase, it can't keep phosphorylating the phosphorylase, but insulin turns on the phosphatase enzymes, and you see the phosphatase enzymes will send the glycogen synthase back to the active form. The phosphatase will send the phosphorylase back to the inactive form, and that's going to stop the process of glycogen breakdown and enhance the process of glycogen synthesis. And insulin will also stimulate glucose uptake by the cells to give you a ready supply of glucose, which can be fed into the glycogen synthesis pathway here. And this applies to all the pathways. We've seen glycolysis and we see gluconeogenesis, but whether you oxidize fats for energy or whether you synthesize fats to be stored or make membranes or whatever, and whether you put fat into the adipose tissue or whether you take fat out of the adipose tissue, it's all the same. It's all the same in terms of what insulin does and what glucagon does and how they work opposed to each other for whatever the needs are. So let's bring in the endocrine system now and see that there's a larger level of complexity here. And that is, in addition to the circulating hormones, glucagon and insulin, there are other hormones that can work as well. Now, the brain is going to constantly sense what's going on inside the body. During digestion, there are sensors in the digestive tract that send neuronal signals back about whether there's carbohydrates or fats or proteins, and the brain can keep track of that. And the brain can know if fuel is coming in, it can help stimulate the release of insulin, or if blood glucose gets a little bit low, it can stimulate the release of glucagon. And so there's going to be a variety of nervous signals in terms of sensing, but also the brain sending signals down to say, I need you to modulate your activity to preserve blood glucose. For example, the brain has a really nice blood glucose sensor. And when the blood glucose level starts to drop, the brain will immediately start signaling the pancreas to release more glucagon, to talk to the adipose to release more fat, but also to the liver to release more glucose to try to increase the blood glucose levels. This will be done very nicely in the neuroscience class and in physiology, the way the endocrine system will work here. But you have direct modulation of tissues either by neuronal signals or by circulating hormones. And the circulating hormones would be epinephrine, norepinephrine, and cortisol. Now we know that epinephrine and norepinephrine are part of the fight and flight stuff, but you also have a lower level of these things that can be produced that can stimulate the tissues to do a little bit of extra activity depending on what is needed here. And we'll talk about those as we go forward. Now the fight or flight mechanism is a special case there, and I would like to introduce that again here. And that is when we perceive danger or perceive a situation that's going to be important, epinephrine will be that main hormone that gets released. And you recall from the cell signaling lecture that epinephrine is going to work on a G protein coupled receptor that works with adenylate cyclase, cyclic AMP, and protein kinase A, very similar to glucagon. And so the effects of epinephrine, there's so many of them on the body, but in terms of metabolism, epinephrine is going to hit these cells and it's going to tell all the cells, you're in danger, we need to liberate as much fuel molecules as we can right now so every tissue has the fuel that it needs to do what is needed to get out of this danger. For the brain, I need lots of glucose so the brain can think fast and do what's needed. For the muscles, I need lots of glucose for very fast ATP production so the muscles can fight or run. And these kinds of things are going on here. So epinephrine is going to bind to these beta-adrenergic receptors. You saw that in the cell signaling lecture. And that's going to give you that adenylate cyclase and protein kinase A activation. So for example, here you see epinephrine. So we talked about glucagon and the glucagon receptor before, but now we're talking about epinephrine and we're talking about beta-adrenergic receptors right here. But it's still G protein coupled, adenylate cyclase, cyclic AMP, and protein kinase A. Its job is to turn on primarily glycogen breakdown to liberate a lot of glucose that can be released into the blood, so there's plenty of glucose in the blood for the skeletal muscles and for the brain, red blood cells, so that we can have maximum performance here. It's like a hammer stroke. That epinephrine hits and the whole system really activates very, very quickly. Now here's an interesting thing. It's a little bit confusing, and we'll cover this more in the glycogen lecture coming up. But liver has a problem. If you recall, liver is really smart when it comes to blood glucose, glucose levels. And when liver says, I've got a lot of glucose, I've already met my energy needs, the liver wants to start synthesizing glycogen, and the liver wants to start synthesizing fats. And during the fight or flight syndrome, when the epinephrine is signaling that fight or flight state, you don't want the liver to use any glucose for any synthetic activity. You want the liver to release that glucose. And so we also have alpha receptors in the liver, and those alpha receptors have a very different signaling mechanism. Here, epinephrine will bind to the alpha receptor. There is a G protein, but that G protein is going to be a G sub Q, and if you remember, those are the ones that give you a calcium response. So here we have phospholipase C. Phospholipase C will work with that PIP2 molecule. IP3 is released from the lipid. That's the head group that gets cut off. IP3 is going to come over here to the endoplasmic reticulum and bind to the IP3 receptor, which is a calcium channel. Calcium comes out of the endoplasmic reticulum, and that calcium now is going to turn on a lot of other proteins. The main thing it does is it interacts with calmodulin, and when calmodulin binds calcium, that calmodulin is activated. And one of the things it does is it's going to bind to a calmodulin-dependent protein kinase that will phosphorylate the glycogen synthase, and it will turn that glycogen synthase off. You see lots of phosphates here. And so all those phosphates really shut this synthase enzyme down to make sure you're not going to use any of your precious glucose to make glycogen during the fight or flight syndrome. The other thing calmodulin will do when it's activated is it binds to the phosphorylase kinase. That's that kinase before glycogen phosphorylase. It will increase the phosphorylation level of the glycogen phosphorylase and make it even more active, which will break down more glycogen and give you more glucose that can be released to the blood. So this is the alpha receptor. We'll see more of that in physiology, a little bit in neuro, but we'll see the alpha receptors are working through that calcium signal to elicit the response that goes on here. We will develop this a lot more in the coming lectures, especially with the glycogen lecture, because glycogen is such an important source of free glucose. It's going to be important during exercise when muscle needs more glucose to meet the muscle contraction and force generation. And so we'll have muscle signals that will communicate the need for glycogen to be phosphorylase to be more active. Of course, we talked about the nerves. The brain can send a message and say, I need a little bit more glucose, or I need a little bit more power, whatever. The nerves can communicate an activation of the phosphorylase enzyme. And of course, we just talked about epinephrine, the fight or flight, communicating down through its pathway the need for more glycogen breakdown. So you see how interconnected these pathways are. So you have glucagon and you have insulin that's affecting glycogen synthesis and breakdown on a day-to-day normal basis, but then all these extra mechanisms are laid on top of that, depending on whether we're exercising, fight or flight, or whether our brain is perceiving some change of state that we need to take care of. And we will develop all of this. So here's a question that is kind of helpful here. Off of an exam recently, and it is, this cartoon illustrates changes in metabolism that occur in response to which hormone? And there's a couple of hormones that are given you here. So when you look at this, the way to decipher this question is to look at the pluses and the minuses and look at the arrows. This shows glucose, and glucose is obviously going towards glycogen for synthesis. Glucose is being sent to fatty acids, synthesizing fats. We see that glycogen's not breaking down. We see that the fatty acids are being sent to the adipose tissue, which are being made into triglycerides. They're not being broken down. We see glucose coming in. So to me, it looks like this is in a glucose utilization mode. It looks like glucose is plentiful. And so what would you guess would be the proper answer for this one? Yeah, obviously insulin is going to be the case. I've also shown the fasting side where there's not pluses, you see glucose coming out. So if you saw that before, so that's a pretty nice way to ask that basic question there. You and your study partner are having an argument about this metabolic state. You say it's the fasting state. Your partner says it's the fed state. Which of the following aspects of the figure prove you are correct? So this is an example of taking the fasting version and putting it into a test question. We're not allowed to ask questions like this anymore because it's not a real good question, but it's a really nice example to go through. So here you just look at your arrows, you look at your pluses and your minuses. You see glucose is coming out of the liver. You see fatty acids are coming out of the adipose. You see skeletal muscles not taking glucose up. You see no effect. So right away you're getting the idea this is probably a fasting example. It's probably a glucagon modality. So which one are you going to pick then? Yeah, all the above are true. Fatty acids are coming out of adipose. The liver is degrading glycogen. The liver is synthesizing glucose, and the liver is releasing that glucose out into the blood. One more. Why do blood insulin levels stay low following a protein-only meal? Take a look at these and see what you think. Yeah, okay. Hopefully you picked answer B. That's because there's little or no glucose to stimulate the release of insulin. Protein-only meals, not much glucose. The pancreatic beta cells really don't sense the blood glucose level going up, so they don't see the need to release insulin. You may say, well, protein, amino acids, that gives you a little bit of insulin, right? And you're right, yeah, amino acids are a minor modulator, but nothing like glucose. So there's only a small change in insulin that really isn't persistent after a high protein meal, as you saw a couple slides back. The wrong answer is amino acids stimulate glucagon release. Well, they kind of do, and glucagon is going to have an opposite effect on insulin, so that was really not the reason. It's the way it is before fasting, before eating. Fatty acids do not stimulate insulin. Well, that's true, they don't, but the thing here was a protein-only meal, not fats. And then glucagon-dependent pathways are active following a high-protein meal. Well, that's true, but that doesn't say why insulin doesn't come out. Insulin doesn't come out because there really is no glucose in that meal to stimulate the production of insulin. Thank you.